In this video clip, I will show you how to perform meta-analysis and subgroup meta-analysis in R statistics with an outcome that is continuous and an outcome that is binary, and how to perform proportional meta-analysis and how to produce and estimate the effect size for a continuous outcome based on the standardized mean difference, and how to calculate the effect size based on correlation coefficient for a continuous outcome, and how to estimate the effect size based on odds ratio or risk ratio for a binary outcome, and how to generate forest plot and funnel plot in R statistics. If this video is useful, subscribe to the channel and leave any comments or queries regarding the video in the comment section, and click on the notification bell to receive the most recent releases of video clip to the channel, and click on the like icon and share the video with others, so that the channel can develop and improvise to include further wide scope of different statistical method and how to perform statistical techniques in several different statistical packages such as SPSS, Excel, Stata, Jamovi, R statistics, GraphPad Prism, G-Power, GMP, and revaman. Meta-analysis with a continuous outcome based on estimation of the effect size, which is a standardized mean difference that can be calculated based on Hedges G or Cohen D or Glacis delta in R statistics, and generating a forest plot and a funnel plot. So what is meta-analysis? Meta-analysis is a statistical method that combines data extracted from multiple independent studies that addresses the same scientific research questions. The studies are designed to have two groups, intervention group or treatment group and a control or a reference group, and the outcome is measured, in this case continuous, and the overall or the pooled estimate of the effect size, which is the SMD, will be calculated. The meta-analysis also will examine the heterogeneity between the results from these several independent studies by performing a heterogeneity test based on the chi-square and generating an I-squared value that is represented in percentage, with a higher I-squared value indicates higher heterogeneity and low value means lower heterogeneity. Meta-analysis also will generate a forest plot that displays the summary of the meta-analysis, in which the pooled overall effect size, the SMD, is calculated. The heterogeneity result will be also produced, and 95% confidence interval, and a weighted percentage, and a p-value. The plot consists of three sections: descriptive analysis, diagrammatic representation, and overall statistical analysis. Meta-analysis also will produce a funnel plot which examines the publication bias. To perform meta-analysis on a continuous variable or continuous outcome based on an estimate of the effect size, which is the standardized mean difference, launch the RStudio and then load the file that contains the data by clicking on File, then scroll down to Import Data Set and then select from SPSS as the file is saved in SPSS format. Then click on Browse, locate the file and then select the file and click on Open, and then click on Import, and the data are imported into RStudio. Before performing the meta-analysis, load the meta packages. If the meta packages is not installed, then install that package, and then in the section here type library to load the meta package, library meta, and hit enter, and the package will be loaded into R. From the table above as shown, there are here a data extracted from 10 studies, from study number 1 to study number 10, and in each study the participants were segregated into 2 groups, a treatment groups and a control group, and the outcome was measured in a continuous scale by calculating the mean for each group, for the treatment group and for the control group. As shown here, the variable here indicates NT, which means number for the treatment, the mean for the treatment, the standard deviation for the treatment, the number for or the size sample for control, the mean for control, and the standard deviation for the control. So here 2 groups with the total number of participants, the average for the group, and the standard deviation for the group. And to perform meta-analysis and to calculate the overall effect size, which will be here the standardized mean difference, we'll stick with the Hedges estimate of the effect size. In the section here, let's generate an object and call it X, and then use the assignment operator, and then type metacon for continuous and select the command or the function to perform meta-analysis on a continuous outcome, and inside the bracket type data, and then insert the file or the data name, which is here is the copy. Select copy and then insert by paste the file, which is smd meta-analysis, then insert comma and insert the variable 1 by 1 for the treatment, then for the control by typing nt, which is capital letter case sensitive, and as R is case sensitive, then mean t comma sdt comma nc comma mean capital C, and then SDC, then insert a comma space. So here we had inserted the variables that are required: nt, mean t, and sdt, nc, and mean for control, and sd standard deviation for control. For using a statistical analysis method, we're going to type method and then dot sm. So SMD, standardized mean difference. We could use the Cohen D or Hedges G or Glacius delta, but I will use the Hedges G, and I'm going to type only H for Hedges, a capital letter, and then insert a comma, then type S for the summary, and we want the summary to be in the format of SMD, standardized mean difference. Then after that insert a comma. Then also in the analysis or the output analysis, insert the study ID by typing stud lab for study label equal to the name of the variable here, which is a study. And for the effects model, I'm going to use the random effects model by typing random and then give it a T for true, and the fixed will be false for F. And if I hit enter to view the output analysis, just type X, enter, and as shown here in this section, the number of study is equal to 10. K is equal to 10, the number of observations 389, and the random effects model was selected, and the standardized mean difference was calculated and found to be 0.72, and the 95% confidence interval calculated, and the p-value which is less than 0.05. And also a heterogeneity test was performed to quantify the variation with an I-squared calculated equal to 13.1%, which is a low heterogeneity, and a 95% confidence interval for that value. And a test of heterogeneity also produced a p-value that we needed to be greater than 0.05 with a degree of freedom 9, and a Q statistics based on chi-square, which is equal to 10.35. To draw a forest plot, let's clean this. In this section, type forest and then open a bracket, and then insert the object, which is X, and hit enter. Simultaneously a forest plot is generated as shown here. If we enlarge, as shown here in the forest plot, there are three sections: the descriptive, statistical descriptive, the diagrammatic representation, and the overall estimate of the effect size and the heterogeneity test. As shown from the forest plot, the studies are inserted, study number 1 to 10, with experimental group and the control group, and the total and the mean and the standard deviation for each group is inserted, and diagrammatic representation or reflection of the descriptive statistics with the standardized mean difference is calculated for each individual study, and a 95% confidence interval is calculated for each individual study with a weight percentage, which is a reflection of the sample size. Higher weight percentage reflect higher sample size. And in the middle section here, the squares represent the effect size, which is the SMD, and there is a variation in the size which reflects the sample size. Larger squares have larger sample size. The horizontal line here represent the limits upper and lower for the 95% confidence interval, and the dashed line here is the line of overall effect size, and the vertical line here is the line of no effect, and here at the bottom is the scale, and on the on this left hand side is the section for the overall statistical analysis calculation of the SMD based on the Hedges effect size, with a value generated for the pooled or overall SMD, which is 0.7 with 7.2 with a 95% confidence interval, and a heterogeneity test with an I-squared and a p-value. To perform few edits here by reducing the decimal on the control and the experimental for standard deviation and the mean, and also to change the color of the square and the color of the vertical line and the color of the diamond shaped here, which represent the overall SMD or overall effect size. Close the window for the plot, and in the section here type forest again, open a bracket, and then insert the object comma. To change the color of the squares, type COL and then square, and then let's give it a color of red, and then hit enter. Spontaneously the color of the squared are changed into red as shown in the plot. Close the window here, and then to change the color of the diamond from gray into black, select the previous command and then insert a comma, and then type color D for diamond, select the diamond, and let's give it a color of black, and then hit enter. And if we click on zoom, the color of the diamond is changed into black. Close the window here, and then on the command section copy the command and then place it here, and then let's change the vertical dashed line into a blue color, which is the overall effect size, by typing COL and then command, hit enter, and then let's give it a color of blue and then hit enter. And if we click on zoom, you can see the color of the dashed line is blue. To reduce the decimal point for the standard deviation and the mean for both the control and the experiment, close the window and copy the command and place it again here, and then insert comma and then type digits equal to, or digits dot mean equal to 1, and digits dot standard deviation equal to 1, and then hit enter and back again to the zoom. The decimal has been changed for the control and the experiment. So this is the way to generate a forest plot that will summarize the outcome from the meta-analysis and will generate an overall effect size, which is the SMD, which is the Hedges here, and test for also and the value for heterogeneity or variation among the results from several independent research studies. We could also generate a funnel plot by closing the window here and then in the section here type funnel, open a bracket and insert X and then hit enter, click on zoom. A funnel plot is generated, which is a scattered plot. On the x-axis is the plotted the standardized mean difference for each individual separate studies, represented by gray circles, and on the y-axis is the standard error, and the scatter plot is in the shape of a funnel, and the line in the middle represent the overall effect size, which is standardized mean difference, which is around 0.72. For lack of publication bias, the distribution of the effect size, which is the SMD, should be equally on both sides of the line here. So there should be equal number of standardized mean difference. Here we have 1, 2, 3, 4 on this section, and 5 on this section, and 1 outside the funnel. So from this funnel, it is possible to determine whether there is a publication bias. The finding can be validated by performing another statistical test, which is the Hidges regression test, that will assess the publication bias. Meta-analysis with a continuous outcome based on an effect size, which is correlation coefficient, abbreviated R, in R statistics, and generating a forest plot and a funnel plot. To perform meta-analysis using an effect size which is correlation coefficient, launch the R Studio, and then if you had not installed the meta package, then install that in R. And I'm going to load the meta-analysis package by typing library, open a bracket, and then type meta, and this will load the package into the R and ready to be used. Then load also the file that contains the data to perform meta-analysis, and the data are scored based on an effect size, which is a correlation coefficient. To do that, click on file, scroll down to import data set, and then select from SPSS, as the file is saved in SPSS format. Then click on browse, locate the file, and then click on it, which is correlation meta-analysis. Click on open, then click on import. The data are loaded into R Studio, and as shown from the table here, there are 12 studies from 1 to 12, and the effect size estimated is the correlation coefficient, which is R, and a value is scored for each study, and a total number of participants in the study is also recorded. To perform the meta-analysis, then meta-correlation function will be used. First, let's generate an object and call it X, and then use the assignment operator, then the command or the function for the meta correlation for correlation. Click on it, and then in between the bracket type data equal sign, and then insert the name of the file that contains the data, and then insert a comma. After that, place the effect size, which is the correlation coefficient, and it is given an R label, comma, and then the total number, which is given a label of total for the total variable, and then comma. After that, type the summary SM equal to open a quotation mark and then type core for correlation, and then select the effects model, and I'm going to use the fixed effects model. So insert comma after the quotation mark here, and then type random equal to false and fixed equal to true. Insert a comma and then insert also the study ID for each study in the meta-analysis by typing studlab equal to study ID as given here in this variable. And if you hit enter and then type X to view the output of the meta-analysis as shown here, the number of studies are K12 and the number of observations 364, and the common effect model is selected, and the correlation coefficient is calculated to p.5, and 95% confidence interval is calculated for the effect size, and a p-value is also calculated, which is less than 0.05, and a heterogeneity test performed, mainly done on Q statistics, and an I-squared is generated, which is equal to 8% with a 95% confidence interval. And from the test of heterogeneity, the degree of freedom was 11 and the p-value was greater than 0.05. So this heterogeneity test shows that there is low level of heterogeneity between the results. To generate a forest plot, just type forest, open a bracket and then insert X, comma, and then use one of the layout that already pre-installed, and I'm going to use the revaman layout and hit enter. Then click on zoom here, and as shown in the plot for forest plot here, this is the forest plot. As shown, there are three sections. The first section is descriptive statistics, the second section is the diagrammatic representation of the descriptive statistics, and the third one is the overall effect size and heterogeneity test. As shown, the study is inserted from study 1 to 12, and the total number or the sample size is inserted with a weight percentage as given here, which reflects the influence of the sample size on the overall analysis of the meta-analysis, and a correlation coefficient is calculated for each specific study with 95% confidence interval with an upper and a lower limits. The second section here is the diagrammatic representation of the first section. The square symbol here represents the correlation coefficient for each specific study, which is color coded red, and the size of that square or squares does varies depending on the sample size or the weighted percentage. Higher sample size will have higher square. The horizontal line represents the 95% confidence interval with the upper and lower limits, and the vertical line, dashed line, represent the overall effect size or the overall correlation coefficient. The diamond shaped here is the estimated effect size, which is the pooled correlation, which is equal to 0.5. And the third section here is the overall effect size and the heterogeneity test, which is calculated based on the I squared, as it is 8%, and p-value also is generated. So this is the forest plot that is produced from the meta-analysis based on an effect size, which is the correlation coefficient that combines the data from all these 12 studies and pool them to calculate overall correlation. To also produce a funnel plot to check for the publication bias, close the plot, and in the section here type funnel, open up brackets, and then type x, which is the object generated. A funnel plot is produced, and as shown from the funnel plot, it is a scatter plot with an x-axis have the correlation coefficient for each specific study represented as a gray circle, and the y-axis is the standard error, and the scatter plot is in a shape of a funnel with a line in the middle which represent the overall correlation coefficient, which is 0.5. For lack of publication bias, the distribution of the effect size should be symmetrical between the two sections here, or between the upper and the lower part of the graph. We have here on the right hand side, we have 1, 2, 3, 4 correlation coefficient or effect size. On the left hand side, 1, 2, 3, 4, 5, 6, 7, 8. From this funnel plot, it is clear that the distribution of the estimate of effect size is asymmetrical, and there is a bias of publication. This has to be confirmed by additional statistical analysis, which is the Hidges regression test. Meta-analysis in R statistics with a binary outcome based on the effect size, which is the odds ratio, to generate a forest plot and a funnel plot. Binary outcome is a variable that is categorical, a variable that have two levels in it, such as alive or dead, yes or no, smoke, doesn't smoke, have disease or doesn't have a disease. To perform meta-analysis for a binary outcome based on effect size, which is the odds ratio, in R statistics, launch the R Studio and then load the file that have the data. And the way to do that is to click on file and then select import data set, and since the file is stored in an SPSS format, then select from SPSS, and in the new window here, click on browse, select the file that contains the data for meta-analysis, click on it, and it will be loaded into the R Studio as shown here. And then if you tick on import, it will be loaded into the R Studio. And as shown here in the table, there are 12 studies as shown in the left column for the study ID, 12 independent studies, and there are 2 groups in each study. So the participant has been organized into an experimental group for abbreviated E, and a control group, which is C. So here a drug is administered to two groups. One is given the drug, the other one is a control that is given a placebo, and the success rate of recovery from the condition is recorded, and a score is given by an event. So those who succeeded in recovering from the condition are scored, and a column is, or a variable is generated called event in the experiment, and then the total participant in the experimental group is given here, and the event occurring in the control group without intervention is also recorded and inserted in the variable called event in the control, and the total number of participants in the control group is recorded. So there are two groups in each study, and the objective is to do meta-analysis on these data from these 12 studies. And since the outcome here is binary, whether the event has occurred or the event has not occurred, whether the participant has recovered from the condition or not, so it's a binary outcome, then the effect size measured or estimated here will be the odds ratio. And the way to do binary outcome meta-analysis in R statistics is first to install the meta package and then load the meta package into the R statistics. So we'll type here in this section library. I had already installed the package, so I'm going just to load that, and if I hit enter it will be loaded. The next step is to use a certain command to perform meta-analysis. Let's first generate an object, call it X, and use the assignment operator, and then type in the command or the function for the meta-analysis, which is meta pen for binary, and that is the function is already installed there. Open a bracket, and within the bracket, after the first bracket, type in the data name equal sign, and then the data file or name is already generated here, so I'm going to copy and paste that to here. And to continue with the command for meta-analysis for binary outcome, we had inserted the data label or the data name. The next is to insert the variable for the event in the experimental, which is event case sensitive event E, and then comma, and then the total number of participants in the experimental group. So total, it has to be capital, total dot E, and for now this is for the experimental group. So we have the first variable, which is event, then the second variable, which is the total, and for the control group also event here has to be capital letter as R is case sensitive. Then we need to insert the event dot C comma. So this is the third variable that have the event scores for the control, and then the total number of participants in the control group, and that is total dot C. Then we need also to choose the effect model, which I'm going to use the random effects model. So I'm going to type random equal to true, and then for the fixed equal to false. So here I am selecting a random effects model. If I want a fixed effect model, then I will give the fixed true and the random F for false. After that, insert a comma and then choose the method to perform the meta-analysis by typing method equal sign, and I'm going to use the MH method, which is the Mantel-Haenszel method, then followed by a comma. And for the summary of the result, I'm going to type SM, then equal, open a bracket, and I'm going to use the effect size, which is the odd or odds ratio. We could use risk ratio, risk differences, and so on. Then insert a comma, and I'm going also to insert the study labels, which is the first column, by typing stud label or study label, and then open a bracket, and then I'm going to select the variable, which is study ID. So this will select the study ID variable. So this is the command or the function for performing binary meta-analysis through typing the command meta-pen and the data, the name of the object created, which is the data, the event in the experimental, the total in the experimental, the event in the control, the total in the control, and choosing random effect models, and then the method MH and the summary OR, odds ratio, and inserting the study label or study ID. Just drag this bracket to here, and then click on enter. To view the output, just type X and then insert as shown here. The output shows that the number of studies K equal to 12 studies, since we have 12 studies, and the number of observation is 1633, and the number of events occurring, which is E, 741. And the next line is showing the random effects model with an OR and a 95% confidence interval, and a p-value is generated here. And also a heterogeneity test is performed and quantified using tau squared, and a value is given with a 95% confidence interval, and an I squared also given in a percentage with the 95% confidence interval as shown here, and the test of heterogeneity with a degree of freedom given here 11, and a p value, and the q which is based on the chi-square. So from this heterogeneity test, it is shown that there are 0% of heterogeneity, as the I squared equal to 0%, and the p-value is greater than 0.05. We need this p-value to be greater than 0.05. So this output or test of heterogeneity showed that there is no heterogeneity among the results from those 12 studies. To draw a forest plot, just type forest, then open a bracket, and let's insert an X, which is the object that we had created, and then a forest plot is generated as shown here. Shows in this forest plot the study ID, the event experimental control, events and total for experimental and control, a diagrammatic representation with the odds ratio, and the 95% confidence interval and the weight and the scale here. To redraw this in a revaman format, just type forest, open a bracket, then x, which is the name of the object created, and then type layout, open a bracket and or quotation mark, and type revaman. So this will produce the forest plot in a revaman format as shown here. This is for revaman. The studies from 1 to 12, the diagrammatic representation is on the right hand side, the odds ratio is given, the MH method, and random effect model is used, and the odds ratio events in each group. If we close that, back again to the forest, and I'm not going to use the revaman, I'm just going to use a forest plot that is a standard, and I'm going to edit part in it. So type forest and then open a bracket x, and then I'm going to add a pooled, so if we just hit the enter and look at the forest plot here, there are few missing values. So the value for the event for the experimental control is not given here, and I'm going to insert that, and I'm going to give a plot which is an X label by calling it, let's say drug A. So close that one, and then type forest again, open a bracket X, and to insert the event for the experimental and the control, type pooled dot event, and then equal true, then comma, and if you just hit enter and look at the graph here, the forest plot, we can see that the event, total event for experimental and for control is inserted here. Close the plot window, and I'm going to give the x-axis a label. So we type here forest, then x, and then copy the same thing as the pooled. Here we need the pooled event, pooled events, and then comma, and I'm going to label the x-axis by typing xlab equal to, let's call it drug A, space drug A, and hit enter. Click on the zoom, and as shown here, the plot is given a x-axis label which is drug A. To do a few editing on the plot here, close the plot and again select the command here, insert, and then after the drug insert a comma, and then to change the color of the squares in the diagram, type color, and then select a square, which is this one, color of the squares, and then open a bracket and or quotation mark and type red and hit enter. Red is misspelled, then let's select the command again, copy, and then insert that here and correct that into red and hit enter, and then click on zoom. As shown in the plot here, the squares for the effect size or the odds ratio are color coded red. To change the diamond color here, or the diamond shaped, which is the overall effect size, into a black color, close the window, again copy the command for the forest plot, and then after the red type color and then diamond, select the diamond function and then open a bracket and let's give it a black color and hit enter. And if we click on zoom, the diamond shaped now is colored black. So looking at this forest plot, it consists of 3 sections. The first section is a descriptive. The second section is a diagrammatic representation, and the third section here, which is the heterogeneity test and the overall effect size. The first column here is the study ID from study 1 to study 12. The second one is experimental, as shown here, the data for the experimental group and the control group, the events in the experiment and the total in the experimental, and the same for the control for each individual study. And if we scroll down to the end here, it shows that the random effect models with the values for all these columns. And on the right hand section here, it shows that the effect size is the OR, which is the odds ratio for each individual study, with a 95% confidence interval for that OR, and a weighted percentage which reflects the impact or the influence of the sample size on the meta-analysis. And at the end here shows that the overall odds ratio for all these 12 studies and the 95% confidence interval. And here is the scale or the ruler from 0.01 up to 100. And in the diagram here, the red squares represent the odds ratio for each individual study, and the size of that square reflects the size of the samples or the number of participants recruited for each study. A higher sample size will have a higher size of a square. The horizontal line represents the 95% confidence interval for each individual study, and the vertical dashed line represent the pooled odds ratio or the overall odds ratio. In the section here also, heterogeneity test has been performed, an I-square is generated and a p-value is generated, and the I-squared is equal to 0% and the p-value is greater than 0.05, which indicates that there is no heterogeneity between the data extracted from all the studies. So this is the way to produce a forest plot. Now to generate a funnel plot, close this window, and in the section here type funnel and then open a bracket and put X and then hit enter, and then click on zoom. As shown here in this plot, this is the funnel plot as it is in a shape of a funnel, and on the x-axis is the odds ratio for each individual study, and on the y-axis is the standard error. The objective of drawing funnel plot is to determine whether there is a bias in publication or not. For no bias in publication, the distribution of the effect size should be evenly located above and below this middle line here, which is a vertical line, and this represents the overall odds ratio. So the odds ratio for each individual study have to be equally distributed in the both side of this line. From this funnel plot, we have 1, 2, 3, 4, 5, 6, 7 on the right side of the line or above the line, and 5 below the line. So there is bias of publication from this funnel, and another test need to be performed, which is Eager's regression test, to confirm that bias does exist. Subgroup meta-analysis with a binary outcome based on the effect size, which is an odds ratio in R statistics, to generate a forest plot and funnel plot. So what is a subgroup meta-analysis? Subgroup meta-analysis is a statistical method that separate or divide the group into 2 or more subgroups, and then perform meta-analysis on those subgroups to generate and calculate the overall or pooled estimate of effect size, which is in this case the odds ratio, and then compare the pooled effect size between these subgroups. So the subgroup meta-analysis can be performed when there is a binary outcome. So what is a binary outcome? Is a variable that have 2 levels in it. It is a categorical variable that have 2 categories in it. So it is dichotomous. It can be either yes or no, life or dead, smoke, doesn't smoke, and the odds ratio is a estimate of the effect size. To perform subgroup meta-analysis with a binary outcome and to use an effect size, which is an odds ratio, launch the RStudio and then also import the file that contains the data for the studies. And the way to do that is to click on File and then scroll to Import Data Set, and the data file is saved in SPSS. So click on it, then browse and select the file. Select the file which contains the data, and then click on Import. The data are loaded into R as shown here. From the table there are a few variables. The first variable or column is the study ID, and there are 12 different studies. The second column is the event, number of events occurring in each study. The third one is the total number of participants for each study, and the fourth one is the event for the control. So C stand for control, E stand for experimental. So the study, each study was designed to have 2 groups, experimental group or exposure group and a control group, and the event is recorded in each group, and the objective of the study is to assess the efficacy of a drug in treating certain condition, and if the condition has been recovered and the participant healed completely from the condition, then it is scored as an event occurring for the experimental or exposure, and if for the control, no treatment or exposure being given to a participant, and if a recovery from the condition happens, then it is recorded and scored as an event in the control. The NC stands for a number of total number of participants in the control group, and finally a section or a column or a variable that have gender in it, male or female. So 6 of the 12 studies, only males are recruited for the study, and for the other 6 only females are recruited. So here we have 6 studies with only males and 6 studies with only females. So here, to find out whether there is a difference in the efficacy of the treatment of the drug between males and females, then subgroup meta-analysis can be performed. And as the data here or the outcome is binary, either the event occur or it doesn't occur, so it is a dichotomous or binomial outcome, then the odds ratio will be calculated and the subgroup meta-analysis will be performed. And the way to perform subgroup meta-analysis in R statistics is to use the meta package. If the meta package is not installed, then install that into the R, and I've already installed the package. Library, open a bracket, type meta, and then hit enter. The library or the meta package is loaded into R, and to do a subgroup meta-analysis for a binary outcome based on the odds ratio, we'll use the command meta pen for binary. And the first thing is to do is to generate an object, call it X, and then use the assignment operator and use the command meta pen, open a bracket, and then insert the data name, which is in this case the file we called it as this one, and then insert that in that part here. So the data is the odd ratio meta-analysis. The next is to insert the events in a sequential event, exposure, n exposure, then event control, n control, and it has to be capital letter dot e, and then comma n dot e, comma, and then event dot c, comma, and n dot c, comma. So here the command is the meta pen, and the data is the file which is given a name, OR meta-analysis. And the first column is the event for the experimental, the second one is the total number for the exposure or experimental, the third one is the event for the control, and the last one is the total number of the participant in the control. To choose a method for the analysis, type method, then insert a quotation mark, and then type MH method for Mantel-Haenszel method, and for the summary view type SM, and then equal sign, quotation mark, and then use the OR. Since we are using here the effect size estimates OR, we could use also risk ratio, but here our objective is to use the odds ratio. If we hit enter here, meta-analysis will be performed on the 12th study or studies. They all will be pooled, and an overall estimate of the effect size will be calculated, and the heterogeneity test will be performed for the 12th study. If we are interested in doing subgroup meta-analysis based on a factor or a variable which is the gender, the meta-analysis will be performed, and the group of the study will be sectioned into 2 groups male groups and females group, and the overall effect size for the male subgroup will be calculated, and for the females will be also calculated with 95% confidence interval and a p-value. And to do a subgroup meta-analysis, insert a comma, then use the command which is by var, select the function which has a equal sign, and then type the factors or the variable to subgroup based on it. In this case is the gender. So this function will perform subgroup meta-analysis for the male group and the females group. To select also a random effect model, then insert comma after the gender option, and then type random equal t for true and fixed equal to false. So here, as I want to use the random effect model, then t is true and fixed is false. If I want to swap, I want a fixed model, effect model, then fixed should be t and the random should be f for false. If I hit enter and then view the meta-analysis or subgroup meta-analysis, type X and hit enter. As shown here from the subgroup meta-analysis, the number of studies are 12 studies, the number of observation 1633, and the number of events 741. And the model is selected, random effects model, and an odd ratio is calculated for the overall estimate effect size with a 95% confidence interval and a p-value. Also, the heterogeneity was quantified by two approaches, the tau squared and the I squared, and the 95% confidence interval is generated. For the I squared, the heterogeneity is equal to 0 and the 95% confidence interval is generated. For test of heterogeneity for the 12 studies, a Q statistic was calculated, which is based on chi-square and a degree of freedom of 11 and a p-value of greater than 0.05. So this result shows that there are no heterogeneity among the results from all the 12 studies, as the p-value is greater than 0.05 and the I squared is 0%. And for the subgroup analysis based on random effect model, the studies segregated into gender or based into gender, male and females, and the number of studies 6 for male and females. The odds ratio for male is given as 19.3 and 15.09 for female. For females, and as shown here, the odds ratio, overall odds ratio for the 12 study is 16, for the male is 19.3 and females 15.1. A 95% confidence interval is generated for each subgroup with a tau squared and an I squared, and as shown here, there is no heterogeneity among studies, 6 studies from the male, and there is a 35.9% heterogeneity between the studies for females. So there is a heterogeneity for the females group, and there is homogeneity for the male group. And the last section here is the test for the subgroup differences based on a random effect model, which give a degree of freedom of 1 and a q value and a p value greater than 0.05. To generate a forest plot, let's type here, let's clear this first, clear this, and then type forest and open up brackets and then x. And if I want to generate a forest plot based on already format such as the ref format for forest plot, then type layout equal signs and then quotation mark revaman and hit enter. And as shown here, if you click on zoom, forest plot is generated for the 12 studies for the even subgroup meta-analysis as shown here. This is the format for the revaman forest plot. In this section is the study or subgroup based on the gender, which is male or females, 6 studies. However, the study ID is not inserted. We will do certain changes on the basic forest to add these. And also there is a section here for the experimental and the control, 2 groups for each study, events and total, and the weight percentage and the odds ratio calculated based on the random effect model with MH method, and a ratio odds ratio with a 95% confidence interval, and a diagrammatic representation of the forest plot. So as shown here, there are 3 sections in the forest plot: the descriptive statistics, the diagrammatic representation, and the overall test, or the overall test and the effect size generated, and heterogeneity test. And as shown here, for each section a heterogeneity value is generated for the male and the females section. So if I want to revert back to the basic format for the forest and I insert and design the format as I would like, then we could use the basic command, which is forest, and then click on open a bracket, hit X, and as shown here, this is the format that is given when we use the forest command. We can see here the study label is missing, the events here are missing for the control and the experimental, and we could also do some editing for the section for the diagrammatic representation, and to add the study label, exit of this view and then back into the command section here, forest, type forest X, then pooled. Let's first of all do the study label by using the study label command equal to, and then the study label is the study ID, so insert study ID. And if I hit study ID has to be capital letter, as the ID has to be capital letter as R is case sensitive. If I hit enter and then click on zoom, the study label for each of the 12 for the male and the female are added here. To add the totals for the event, for the experimental and the control, exit of the plot, and then in the command section type again forest, open a bracket X, then keep copying the study label and insert that here, and then comma, and then type pooled event equal true, and hit enter and click on zoom. As shown here, the pooled event for experimental and control for the subgroups are added, for all the missing data here. To do a few editing for this diagrammatic representation, to change the color of the square here, and also the line, vertical line, which is the overall odds ratio, then exit of the plot, and in the command section select the command here, forest, and paste, and then add a comma, and let's change the color of the square by typing color.square, and let's give it a color of red. And if I hit enter and click on zoom, as shown in the diagram here, the color of the squares are colored with red. To change the color of the diamond here, which represent the overall effect size, exit of this, copy the previous command, insert, and then add a comma, and then type color diamond and select the color diamond option, and then let's give the color of the diamond black, and hit enter, view it again, and as shown here, the diamond is colored black. And finally, to change the color of this line, let's say dotted or dashed line, to blue, close the plot, copy the command, and then paste that here and add to that comma, and then let's type column of the random, since we use the random approach, and then let's call it or give it a blue color, and hit enter, and then click on zoom. As shown here, it is a faint blue color is assigned for the vertical line. So this is some of the editing that can be done on the forest plot manually, rather than having already a pre-installed shape for forest plot based on revaman. So from this subgroup meta-analysis, there are three sections as shown here: the descriptive statistics, the diagrammatic representation, and finally the overall. So from the section here, we see that the studies are sectioned into male and females based on the gender, and the experimental group in each study with the control, the events and the total are counted and inserted here, and the odds ratio for each individual study is calculated with a 95% confidence interval and a weight percentage, which calculates and add to it the value of the size of the sample into the overall effect into the meta-analysis. So higher percentage reflect higher participant number in each study. This is the descriptive statistics. For the diagram here, this is the plot which shows that the vertical blue line now is the overall effect size, which is an odds ratio, which is found to be around which is here around 16.24 for the overall meta-analysis without subgrouping. So this is the blue line, and for the small size squares with different sizes, that represent the odds ratio for each individual study. Larger squares represent larger study with larger number of participants. The horizontal line is the 95% confidence interval. The diamond shaped structure or symbol here reflects the odds ratio for the male subgroup and the one here is the females subgroup, and for the male value is given here, and for the females the odds ratio for it is given here. And as we can see that there are variation, the males higher, females lower, and the overall for the 12 study without segregation is 16.24. This with segregation based on gender, there is a variability in the odds ratio. The third section here is the heterogeneity among the study when they are segregated into male and females, and for the overall effect. As shown here, for the male, the heterogeneity, the I squared is equal to 0, and the p-value is greater than 0.05. So there is no heterogeneity among the 6 studies for the male. For the females, the I squared is 36%, and the p-value is greater than 0.05. So there is a heterogeneity among the results from the 6 studies, which only includes females participant in it. And the last one is the heterogeneity for the overall meta-analysis rather than sub-meta-analysis. And as shown here, the I squared is 0% and the p-value is greater than 0.05. And a test of subgroup differences is also generated here. So from this forest plot, it is possible to look at the overall effect size based on just meta-analysis, or we can segregate the studies into 2 sections, males and females, and then calculate, perform meta-analysis for each subgroup, and then compare the outcome from those 2 groups. This is for a subgroup meta-analysis generating forest plot. To generate a funnel plot is plotted to determine whether there is a publication bias, and to draw the funnel plot is just type funnel, open a bracket X, and then hit enter. And if you click on zoom and drag the graph to this section here, as shown here, a funnel plot or a funnel shaped diagram is generated, and on the x-axis is the odds ratio for each individual study. The odds ratio is the estimate of effect size, and on the y-axis is the standard error or the sample size. So this is a scatter plot with small circles here represent the odds ratio for each individual study. The line on the middle here is the overall effect size. So this funnel plot is for not the subgroup meta-analysis, it's just only for the whole 12 studies. And the distribution of the studies in reference to the overall odds ratio, which is around 16.3 here, has to be equally distributed above and below the line. So here we have 1, 2, 3, 4, 5, 6, 7 on the above section of the value for the overall or pooled odds ratio, and have 5 below. So there is a degree of bias in publication from this funnel plot, and another step need to be done, such as Eager's regression test to confirm that observation. Meta-analysis with a binary outcome based on an effect size estimate, which is a risk ratio in R statistics, to generate a forest plot and a funnel plot. So what is a binary outcome? A binary outcome is a variable that have 2 levels in it, such as agree, disagree, live or dead, smoke, doesn't smoke. So this variable is dichotomous or binomial, as it only have 2 categories in it. The risk ratio is the ratio of the probability of an event occurring in one group, which is a treatment group or intervention group or exposure group, compared to the probability of this events, similar events happening in the control or reference or unexposed group. So it is the risk or the ratio of the risk of an event in one group relative to the risk in a control group. To perform meta-analysis in R statistics for a binary outcome based on the risk ratio, first of all, import the file that contains the data from the studies. And the way to do that is to click on File, scroll down to Import Data Set, and since the data stored in a SPSS format, select that option and then click on Browse, select the file and then click Open, and then click on Import. The data is imported into the R statistics, and as shown here, this is a table that shows data taken from 12 study clinical data for each study for a experimental intervention or treatment. So the participant in this study has been recruited and arranged into two groups: the treatment group or the experimental group or the intervention group, and the control group. And as we can see here, there are 12 independent studies addressing specific question, and this column shows that the experimental events were counted for each group in each study, and the total number of participants for the experimental group is counted, and the same thing for the control or the reference group, in which the occurrence of the event is counted and the total number of participants in the control group is also recorded here. So each study was designed to have two separate groups. One is experimental or exposure and the other one is the control, and the events in each group is counted and recorded. So here we have an outcome that is binary, whether the events occur or it doesn't occur. So it is a dichotomous binomial outcome, and the risk ratio could be calculated and used to estimate the effect size. So to do a meta-analysis on this data based on risk ratio for a binary outcome, first install the meta-analysis package. I've already installed the package, so I'm going to type here at this section library to load the meta package, hit enter, and the package is installed or loaded into the R. And to do a meta-analysis, I will use the command meta-bin for binary. And first of all, let's generate an object and call it X and use the assignment operator. Then type the function for meta-analysis, meta binary, hit enter. Then a bracket will be opened. Inside that bracket, insert the data, which is as here being loaded, which is called the RR event meta-analysis. Copy and paste that in here section. Then insert comma and then after that insert the first event in the experimental group, followed by the total in the experimental group, and then followed by the event in the control group, followed by the total in the control group. So we'll type here the event in the experimental or exposure group is T capital, as the R is case sensitive, and event for the first set of data or variable. Then the next one is the total, T for total, comma, and then select the one which stands for capital letter C underscore, and then event, comma, and then capital letter underscore total, and then insert comma. So to double check, so we select this variable as it has the event in it, and then we select the total number for the experimental or exposure group, and then for control group the event for the control and the total for the control. Then for the meta-analysis we could use random effects model or fixed effect model. I'm going to stick with the random effect model, so type random, then equal sign, and then type T for true, and for the fixed model I'm going to type false. So if I want to have to calculate the meta-analysis based on random effect model, write that in this format: T for random, F for fixed. If I want to reverse that situation in which I want a fixed effect model, then a fixed will be true for T and random F for failure. Insert comma and then choose the method to use for the analysis. So type method and then equal sign, and I'm going to stick with the MH method for Mantel-Haenszel method, then comma, and then for the summary type SM and then quotation mark, and since we are using the risk ratio type RR, and then insert a comma and then insert also the label for the studies by typing study, put the cursor here and type study lab, which is study labels, and then the study label will be the variable which is here, the capital letter study ID, and then hit enter. So the command for the meta bin for meta analysis will use the data that we imported that has the events in it for the two groups, the control and the exposure, and then the event from the experimental and the total, the event from the control with the total and random model, and method is MH, and the effect size is the risk ratio, and the study ID is also added. If I type X to view the output as shown here, a meta-analysis is performed but not yet force plot and funnel plot produced. So the output shows that the number of studies K equal 12 studies, the number of observation 1348, and the number of event 429. Also, as shown here, the random effect model is considered and used, and the risk ratio is generated, which is 2.34, and a 95% confidence interval and a p-value. And also a heterogeneity test is performed based on two statistics, the tau squared is generated with 95% confidence interval, and an I-squared also generated, which shows that 22.3%, and that is a value represent the degree of heterogeneity among the results with a 95% confidence interval, and a test of heterogeneity performed based on the Q statistics, which is based on the chi-square and the degree of freedom of 11 and a p-value of greater than 0.05. So from this data here, it shows that there is low level of heterogeneity among the 12 studies, as the I-squared was 22.3 and the p-value to be greater than 0.05. From this meta-analysis, as shown here, that there is a higher risk ratio, which is 2.3 when we compare the exposure to the control. So this is the objective of doing meta-analysis, is to calculate the overall risk ratio rather than individual risk ratio for each individual study, and the overall is 2.34. The next step is to generate a forest plot. And the way to generate a forest plot, let's clean this. Either I use already pre-installed format, or I can use a basic format and then modify the structure and edit the forest plot. So if I type forest and type forest, open up brackets, put the name X, and then I could use a pre-installed format, which is type by typing layout equal to, and then select the revaman format. And if I click on enter, straight away forest plot is generated, and as shown here, this is in the format of a revaman. There is a 3 sections. The first section is descriptive statistics, the diagrammatic representation of this descriptive statistics, and the overall effect size, which is the overall RR. And as shown here, there is a section column for the studies from 1 to 12, the experimental and the control, the events, the total for each section, the weight percentage, which reflects the size of each individual study, as each study contribution to the overall analysis is weighted and given in percentage. Higher percentage indicates larger sample size. The MH method is used, with the risk ratio is calculated for each individual study with a 95% confidence interval with a lower and upper limit. And the second section, which is diagrammatic representation of the descriptive statistics. The overall or pooled risk ratio is given, which is 2.34 with 95% confidence interval. And the third section is that section that include the overall estimate and the heterogeneity test. We could also go back again and draw the forest and edit it in the certain style you wish to present. If we close the plot and in the command section type forest X and hit enter, that will give me the basic structure for the forest plot. And as shown in the forest plot here, there is a missing value for the events, and we need to add this. And also I could do some changes in the coloring of these diagrammatic representation for the color of the squares and the color of the diamond, and for also the color of the line, which represent the overall effect size or overall risk ratio. So to start with this, we add the pooled event for experimental and control. Back to the command section here, type forest, open up brackets X, and then type pooled dot events, hit enter, and then type T for true and hit enter again. And let's bring the graph or the plot, and as shown here, the total number of events for the experimental and control is given. To change the color of the squares, just type forest, or let's select the previous command here and then paste here. And for changing the color of the squares, insert comma and then type COL for color square and use the first function, and between two quotation let's make it red, and then hit enter and then bring the plot as shown here. The squares are color coded red. To change the color for the diamond, which is by the way, this is a specific risk ratio for each individual study, and this one is for the overall. To change the color of the diamond, back again to the command section, copy the previous command, insert it here, then insert a comma and then type COL dot diamond and select the function for diamond, and in between two quotation let's make it here green. If we type green and then hit enter and bring the plot as shown here, the diamond is color coded green. And to change the dotted line here into blue, again copy the command, place it here, and then after the comma type call dot, since we used the random models, select that equal sign and then quotation mark, and let's make it blue in color. Hit enter, click on zoom, and as shown here the dotted line is blue in color. So this is the way to generate a forest plot. So this forest plot will summarize the meta-analysis for all the 12 studies, and as shown here there are a risk ratio calculated for each individual study and an overall risk ratio from the 12 studies for comparison, and the squares here represent the risk ratio for each individual study, and the size differences is reflection of the size of the sample or the weighted percentage. Higher sample size have higher square. The horizontal line represents the 95% confidence interval, and the vertical line, dashed line, is the line for the overall risk ratio, and the diamond, as I said, is for the overall representation of the overall risk ratio. And the section here gives the heterogeneity test for I-squared and a p-value, and from this meta-analysis a comparison has been generated between the experimental to control, and overall value is generated, which is 2.34, and this value is significant as shown by the 95% confidence interval. We could also generate another plot, which is the funnel plot, and the objective of the funnel plot is to assess bias in publication. And the way to do the funnel plot is just type funnel, open a bracket, and put the x value, which is the object, and a funnel plot is drawn as shown here. The funnel plot has on the x-axis is the risk ratio for each individual separate study, and for the y-axis is the standard error. We could select also sample size if you wish, and this is a scattered plot in a form of a funnel, and the line in the middle is the pooled or overall risk ratio, which is 2.4, around 2.4, and the small circles here are the individual risk ratio for each study. For the lack of bias, no bias in publication, the distribution of the effect size or the RR for each individual study should be equally distributed on both section here, above or and below the line, which is the overall risk ratio. And here we have 1, 2, 3, 4, 5, 6, 7, 8 studies with a risk ratio on the right side and 3 on the left and 1 outside the funnel. There is a bias in publication and an additional test needed to be done to confirm and supplement the finding, which is the Hidges regression test. Proportional meta-analysis in R statistics and generation of forest plot and funnel plot. Proportional meta-analysis is a single group meta-analysis which is different from pairwise meta-analysis. In pairwise meta-analysis, there is 2 groups, one intervention or exposure, and the other one is a reference or unexposed group. In a single group meta-analysis, only one group of data are used. So what is proportional meta-analysis? It is a statistical method that combines results from several studies. Those results or data extracted from multiple independent studies. The effect size estimated or measured in this analysis can be percentage, proportion, rate, or frequency. So the proportional meta-analysis will calculate the pooled overall estimates of the proportion or the rate from those several independent studies addressing specific scientific, medical, or clinical question. So any binary data that is reported in percentage or rate or proportion can be included in the meta-proportional analysis. The method is used commonly for the analysis of the prevalence or incidence of condition or disease in population. The meta-analysis also will assess the degree of variability or inconsistency among the results from those several independent studies by performing Q statistics, which is based on chi-square, and a p-value is generated, and an I-squared, which is represented in percentage. An I-squared of 0% indicates that there is no heterogeneity between the results from those several independent studies, and 100% I-squared value indicates maximum heterogeneity. The proportional meta-analysis also generates a forest plot. A forest plot is a graph that displays the summary of the outcome from the meta-analysis. It will include in it the proportion or the rate, the 95% confidence interval, could be a p-value, a weighted percentage. So the graph or the plot is made up of 3 sections: statistical summary, diagrammatic representation of the statistical summary, and the third part, which is the overall estimate of the effect size or pooled effect size, which in this case the proportion or the rate, and the test of heterogeneity with I-squared value and the p-value. The proportional meta-analysis will also produce a funnel plot to test and examine the publication bias, and the plot is a scatter plot. The funnel plot is a scatter plot in which in the x-axis the estimate of the effect size, which is the proportion or the rate or the percentage for each specific study, is plotted on the x-axis, and on the y-axis is the standard error, and the plot is in a shape of a funnel, and the distribution of the effect size for each studies under the funnel will indicate whether there is bias or no bias in publication. To perform proportional meta-analysis in R, launch the R Studio, and then also load the meta package. If you have not installed the meta package, you need the meta package for performing meta-analysis. I've already had installed the package, so I'm going to load it into the R Studio by typing library, open a bracket, and then type meta library, open a bracket, meta, and hit enter, and the meta package is loaded into the R. To load the file that contains the data for meta proportion analysis, click on File, scroll down to Import Data Set, and then select from SPSS as the file is saved in a format of SPSS. If the file is saved in another format such as Excel or text or SAS or Stata, then choose the right options. I will click on the file from SPSS, and then in the new window browse to select the file. Once the file is located, select the file and click on open, then click on import, and the data is loaded into R. As shown here in the top section in the table, there are 12 independent studies labeled from 1 to 12 with a label of study ID named for that variable, and a second variable which is the event for specific condition, and then the total number of participants or recruited for the study. So to perform meta-analysis, proportional meta-analysis on these 12 studies, in the section here, let's generate an object and call it X, and then use the assignment operator and type meta proportion, as we are going to perform a proportional meta-analysis. So this is the command for single group meta-analysis, which is meta proportion, as I have here only one set of data, and in between the bracket type data equal to and the name of the file or the object that has been loaded into the R, which is this one. Then insert comma, and after that insert the variable that contains the event, which is labeled as event, then insert comma, and then the variable that have the total number, which is total, in this case capital letter total, then insert comma, and after that type method, and I'm going to select here the method which is inverse between two quotation mark and inserting an I symbol for inverse. Then after that insert a comma, and then for the summary type SM equal, then quotation, and then P row for proportion row, then followed by comma, and then to also insert the study ID for each individual study. So we need to use the variable which is labeled study ID by typing stud lab or labels equal to, and then study ID, as it is case sensitive in R, so it has to be entered as it's recorded in the table. Then insert comma. Here I'm going to use the fixed effects model by typing first random equal to F for false, and then fixed comma, and then fixed equal to T, which is true. So here what I have decided is that to use the fixed effect model and not the random effects model. If I want to swap, if I want a random effects model, then I will type random equal to true and fixed equal to false. But I'm going in this video use the fixed effect model. So this is the command to perform meta-analysis, proportional meta-analysis. The way to do that is to generate an object, call it X, then use the function which is meta proportion, open a bracket, type data, what is the name of the data, which is this one, then comma, then insert the first variable which is event, then the next variable which is total, then the method which is an inverse, and the summary which is the proportion row, and a study label is inserted also with a fixed effects model chosen. If I hit enter to view the proportional meta-analysis, just type X. As shown here, there are number of studies shows that there are K equal to 12, and number of observations 1212, and the number of event 426, and the model used here is the fixed, it's called here common effect model, and the proportion is calculated. This is the overall or pooled proportion, which is 0.34, and the 95% confidence interval, and the proportion is within the lower and the upper limits for the 95% CI. Also, a heterogeneity test was performed and an I-squared is generated, which equals to 70%, and the 95% confidence interval for that value is also generated, and the test of heterogeneity performed using the Q statistics with the value produced, and degree of freedom and a p-value less than 0.05. To draw a forest plot, just type forest and then open a bracket, insert the object which is X, and if we want to draw a forest based on a layout by revaman, then type layout quotation mark and then revaman and hit enter. A forest plot is generated in the format of revaman as shown here: the study IDs, the events, the total, the weight, the model effect which is the fixed, and the 95% confidence interval, the diagrammatic representation, the scale, and the overall values. However, if we want to do a forest plot and draw a basic forest plot, edit the plot as we wish, then close that plot, and in the section here type forest, open up brackets and then type X, and a basic forest plot is generated. As shown from this plot, the study ID is inserted from study 1 to 12. The events are added here and the total for all the 12 studies are calculated. However, the total for the events is missing here, and in the middle section is the diagrammatic representation, and the proportion is given here, and the 95% confidence interval with the weight percentage, and an overall effect size, which is a proportion, and a heterogeneity test. If we want to add the total number of events to the plot, close the plot, and in the section here type forest, open a bracket, x, comma, and then type pooled events equal t for true, and hit enter, and then click on zoom again as shown here. The total event is added here. To add a label here for the diagrammatic representation, close the plot and then copy the command for the forest, insert it here, and then comma after the true, and then type sm summary label equal to, and let's give it a name which is proportion. The proportion word has to be between two quotations marks, so type proportion and then hit enter. Click on zoom again. As shown here above the diagrammatic representation of the forest plot, the proportion label is added. To change the color for the squares and the diamond and the vertical line here, the square here represent the proportion for each individual study. The diamond represent the overall or pooled proportion from the proportional meta-analysis, and the vertical line represent the overall proportion. To change the color for the square symbols here, close the plot, and in the command section copy the command for forest, and then insert that here, and then insert a comma. And to change the color of the square symbol, type col dot square equal to, and let's give it a color of red, and then hit enter. Click on zoom, and as shown here, the square the effect size for the proportion, which is the square, is changed into a red color. To change the color of the diamond into a black color, exit the plot, copy the previous command and insert that here, and then after the quotation insert a comma. To change the color of the diamond, type col dot diamond, select the color dot diamond, and then equal sign and quotation mark, and type black, and then hit enter. And as shown here, if we click on zoom, the diamond color is changed into black. To change the color of the vertical line, close the plot, copy the previous command, insert that in this place here, and then insert a comma, and then type col dot common, and open a quotation mark, and let's color code it blue, and hit enter. And if we click on zoom, we can see that the vertical line is colored blue. could also change the scale in this ruler here from 0.1 to 0.7, and the way to do that is to close the plot, copy the previous command, copy and then insert here, and then after the quotation insert a comma and then type x limits x lim equal to, and then insert c. The lower value will be 0.1 and the upper one will be 0.7. If we hit enter and then click in zoom, as shown here, the scale is redrawn from 0.1 to 0.7. So this is the forest plot. From this forest plot we can see that there are three sections: the statistical summaries, the diagrammatic representation, and the overall statistics. Here, as shown, the studies are inserted from study 1 to 12 with the events in each study and the total of participants in each study, and the proportion is calculated for each independent study with a 95% confidence interval and a weight percentage, which reflects the sample size, as higher samples that have a larger number of participants will have a higher weight, as the weight can influence the meta-analysis. The second section here, which is a diagrammatic representation of the descriptive statistics. The red square here represent the proportion for each individual study, and the size reflect the sample size. The size of the square reflects the sample size. The horizontal line represent the 95% confidence interval with the lower and the upper limits for it. The vertical blue line is the represent the effect, overall effect size or overall proportion, which is 0.34. And the third section is the overall statistical analysis, or statistical analysis for the overall estimate effect size and heterogeneity. As shown here, the overall proportion is 0.34 and 95% confidence interval is calculated. And for heterogeneity, the I-squared is 70% and the p-value is less than 0.05. So this forest plot shows that the 12 studies, or the data from the 12 studies, were pooled, and the overall estimate of the effect size, which is the proportion, is calculated. To draw a funnel plot to check for publication bias, close this plot, and in the section here type funnel, open a bracket, and then insert the object, which is x, and hit enter. Click on zoom. A funnel plot is generated as shown here. This is a scatter plot with the x-axis represent the proportion for each individual study as indicated by gray circles, and on the y-axis is the standard error, and the shape of the plot is similar to a funnel. The line in the middle here represent the overall proportion, which is calculated to be about 0.34. And for lack of publication bias, the distribution of the effect size, which is the proportion for the studies, has to be equally distributed in both sides of this line. So it has to have symmetry in the distribution. From this funnel plot, the publication bias can be assessed, and to further validate the finding of the funnel plot, then Hidges regression test can be performed. If you are an undergraduate or a postgraduate or even a researcher and you have a data and you want to analyze that data in any of these packages, statistical packages, such as SPSS, Stata, Jamovi, R-Statistics, GraphPad Prism, GMP, or revaman, then contact me to arrange a one-to-one private tutor session. Or if you are interested in joining a training course for performing statistical analysis using any one of these listed packages, then do contact me on the following email address to arrange for either a private tutor session or a training course for several weeks.